Thank you very much. So I'm going to change the rules. You could just call me Yumi to keep it simple. And I've been kind of thinking about the whole purpose of this gathering and stuff like that. And yeah, I could really focus on the world of how to get a molecular test going from a research lab to a clinical setting. But what I really want to emphasize and present to you is the philosophy of what does it take to do a scientific experiment with samples from a human being, from a patient. And basically, this is the world of really translating the science that you guys are working on and putting it to so-called clinical or practical use. So I'm just going to use as an example of my experience taking molecular biology assays from an academic setting and what it takes to do it so that the government, health clinics, insurance companies is willing to pay for you to perform these very complex assays. Okay, so my background is I have a PhD in molecular biology, and I did do a postdoc in fruit flies due to the inspiration of basically dating him at the time. So I actually did a postdoc in Drosophila melanogaster and got to really understand the promoter of a gene that's expressed in the embryo. But I did not want to stay in the academic setting. I wanted to take my knowledge and my thinking that I learned from the PhD and the postdoc and put it in a more applied setting. So what I ended up doing is I got a position for a very large pharmaceutical company in St. Louis, Missouri, using my expertise in molecular biology to help identify new targets for drug. This is the first time they were trying to approach precision medicine and try to understand, make a drug for a particular enzyme, and therefore you will be able to treat and create that $1 million pharmaceutical drug. But that lasted 10 years. And then basically private industry, it's fantastic to work for, but business decisions are made. So they shut down the institution. Oh, sure. Okay, wasn't me. So they shut down the institution that I was working for, and I transitioned over to be a project manager at the Washington University. It's now known as the McDonald Genome Institute. This is one of the 3 major centers that help publish the human genome sequence. And when I joined them, they were transitioning from, okay, we now know the so-called sequence of a reference human being. Let's apply it and start identifying the different sequence variations in a particular patient with this type of clinical phenotype. What variations occur to that? So in other words, it's resequencing an individual or different individuals over and over again and trying to correlate the human variation with a clinical phenotype or what have you. So that's why I have there human gene resequencing. Oops, sorry, wrong button. Human gene resequencing. But it was in that setting that even though it was an academic institution, it was being run like an industry. They had a huge staff, lots and lots of sequencers, and that's where it was a kind of a lovely transition from going from a small academic lab, large pharmaceutical company, and now to a large academic center where they were cranking out sequence over and over and over again. And then I followed Ross, actually back to our hometown of New York City. And I ended up in the Department of Clinical Molecular Genetics at Mount Sinai. And this was another fortuitous opportunity. I landed in a department that was half academic research, a setting I was very familiar with, but also half clinical. They actually had clinical geneticists, MDs, genetic counselors, and more importantly, they had laboratories that were so-called accredited because they were taking human samples, performing complex scientific experiments or molecular assays, generating a result that would go back to the patient. The result would go back to the doctor. It would go back to their medical record and have an impact on the type of therapy that they would have. So this is what I mean by a clinical setting, right? And from there, I became board certified in clinical molecular genetics, which is basically saying that I have experience. I got to see what it's like to basically work with patients. I got to see what it's like, most critically, in a lab that has these rigorous controls in place. And that's what the bulk of my talk is. And from there, I became a certified lab director, and I did a very American thing of climbing the career ladder, which is not staying with one organization over the entirety of my career. It is actually going from one organization to a different organization to a different organization. And each of these changes basically gave me either new responsibilities, more responsibilities, put it bluntly, salary increases. It allowed me to climb the so-called career ladder. And then ended up, again, we decided to go for a big move, which is to move to Scotland, in particular Glasgow. And I originally got hired by a small company called BioClavis. They're a very small biotech company, and their goal is that they wanted to take their unique molecular assay and get it so that it can be recognized and used by the NHS or used by, proved to be used by insurance companies or what have you. So they wanted me to bring my laboratory, clinical laboratory experience. What does it take to get a test so-called accredited, a laboratory accredited, and put it to use? And what happened at the time is that we moved to Glasgow when the whole world shut down. We literally moved that weekend of the UK went through the COVID lockdown. We left New York City that same weekend. They went through the COVID lockdown. So we landed in Glasgow, and we basically stayed with friends, colleagues for 4.5 months during the lockdown. And at that point, my boss, BioClavis, he called me up and he basically said, hey, we're locked down. Nobody can do the science. But right across the hall from us, they are building one of the 3 major COVID testing centers for the United Kingdom. And basically, my boss and I were hired kind of like super consultants to help run this Glasgow COVID testing lab. And again, I was able to bring my clinical laboratory experience to make sure that we generated the right results for the right patient. And on a slow day, we were processing 60,000 samples in a single day. And that was because it was running 24 hours a day, 7 days a week. We had 4 shifts of scientists and all the logistical people, 4 shifts of them. There were also, alternating shifts. So that's allowed us to do that 24-hour time span. And I had an active role in basically making sure that the quality of our results always stayed consistent, so we could send it back for the people who did the nasal swab. Do I have COVID? That type of stuff. So it was an incredible experience because, again, it was something I was familiar running a lab, but it was on a scale that I never had to deal with before. And then from there, basically moved from a different organization, and I'm currently the lab director. I'm back to a very small biotech company that would like to get their test accredited. Okay. So what does it mean to be accredited? So I'm starting off with the United States because it is what I'm most familiar with. And in the United States, the shorthand for accreditation is following the rules or the guidelines presented by the Clinical Laboratory Improvement Act. So basically, the federal government said, if you want to be a laboratory that is performing a patient test, you need to meet our recommended guidelines. And it's gotten to the point, if your laboratory does not meet those guidelines, then insurance company won't deal with you. Your reputation is much lower. And so therefore, CLIA is kind of like the shorthand name. I want my lab to be CLIA. And so in my particular case, I'm more focused on genetic testing. And that was because at Mount Sinai, I got trained by a genetic testing lab. So it's people with like cystic fibrosis, or they might be a carrier for Tay-Sachs disease. Basically, it's tests of the human DNA sample and then trying to find the sequence variation that's associated with it. And then there are many other accreditation groups in the United States. And then here, accreditation, and I'll go into what does it mean to be accredited. It's basically a philosophy of how do you do the work in a laboratory. And I mentioned CLIA is kind of like the guidelines published for laboratories located in the United States. All countries have their guidelines. And most of the time, it's decided which accreditation does my laboratory go for. And the first one is where is your laboratory located? Is it in the United Kingdom? Is it in Nigeria? Is it in France? So for most of the world, they follow the guidelines that's been published by a consortium, ISO 15189. And it's gotten to the point that almost all the countries outside of the US follow ISO 15189 recommended guidelines to run a laboratory. And I was very relieved and kind of like, yes, this does make sense. Their guidelines is like CLIA. We're all following the same standards, the same type of approach and philosophy. They just have different letters, name, acronyms in front of it. So ISO 15189 specifies the requirements for medical laboratories, and that's what we're using in the UK. And we took a quick peek. It's also being used, ISO 15189 guidelines are followed in Nigeria too. And then you can publish the guidelines, and there are different ways of saying, how does the lab demonstrate that they're doing it? And most of the labs, you actually go to a third party, and basically a neutral party. And in the United Kingdom, it's actually the United Kingdom Accreditation Service, or UKAS. They actually they're their own nonprofit organization. They have their own assessors. And when we say, oh, our laboratory is ready to be independently assessed to demonstrate, yes, we are following the guidelines, the UCAS sends their inspectors down to inspect basically what you have done. And that was one of the things that we did with the COVID testing lab. We actually achieved ISO 15189 accreditation. There's a whole world of guidelines. There's a whole world of accreditation. So I just mentioned a small number here. So there's GCLP, which is Good Clinical Laboratory Practice. That is the type of guidelines you would follow if you're participating in a clinical trial. So it's not something that insurance will pay for, but big companies, if they're doing a big pharmaceutical test, and they want a laboratory to perform some type of testing alongside with their big clinical trials, they would want that laboratory to be GCLP accredited. You also have accreditation for manufacturing. So if you want to manufacture your drug, your new therapeutic, you would tend to follow GMP, or Good Manufacturing Practices. And then there's a lot of different ISOs that kind of, could cover the so-called realm that your laboratory is working on. And then there's even guidelines accreditation strictly for IT. This is ISO 27001, which is your IT infrastructure, are your computers secure? Can you demonstrate that you won't be leaking out private information, to the world? How easily can you be hacked? So there are guidelines literally for everything. Okay, so what is the underlying philosophy? So what I'm showing here under the green arrows is a basic workflow that happens in the laboratory, all right, in a research setting. You have your samples, it could be fruit flies or what have you. Oh, by the way, This slide deck will be shared with everybody, so you don't have to take pictures of it. Okay, so you have your samples. It goes through a laboratory. You make data, and that data is used for your publication, for your clinical trial, for your grant writing. This is research. Now we're going to the clinical setting. The same process is happening in the laboratory, samples, laboratory data, but the key difference is the sample that's coming in is coming in from a patient. So it's not like, oh, we have 200, samples coming in and 95% of them have this type of molecular characteristic and 5% of them have that. No, it's patient one. Their sample goes to the laboratory. It produces data only for patient one. And that result goes back to the doctor, the patient, the hospital. Okay. And so that is the key difference. Now we're concerned about that one individual sample. All right. Not a population of samples. All right. So what happens in the laboratory? You have the lab space. You have a process, your protocol that you want each person to do over and over and over again. You have equipment. It could be sequencers, it could be mass spec, it could be a gel, or real-time PCR, as you'll get to experience. You have reagents, the chemicals that actually do the work that you are purchasing from a vendor. You have software. Everything is done on computer. It could be an Excel spreadsheet. It could be a powerful database where you're performing, back in the day, it used to be done on supercomputers, but now it's done up in the cloud. And most critical, you have people, human beings who are actually doing the work. So the underlying philosophy in a clinical setting is, what do you do to minimize error? Because it will impact that one patient. So focusing on that orange flow where it's all about the clinical space, everything is about minimizing error. So what do you think about, how do I minimize any type of error? You ask things like, in the lab space, is the space safe? Is it secure? Is it comfortable? Will the machine work in that space? So I've heard people where they started off their lab in a container and they would walk in the morning. This is in the UK. Their reagents are frozen. Okay, you can't do work that way. So even asking things like, is the space secure and under the right conditions that will allow you to perform your work? Protocol, does the protocol work? So in a research setting, you got your assay, you're following, you're adding buffer A, buffer B, you're pipetting this volume. It's very easy to say, oh, I know something that'll make it work better, and then immediately change it. In a clinical setting, you do that, but in a very controlled fashion, because all of a sudden you've got some people following one protocol, generating one type of results, and somebody else following a completely different protocol. So one thing is, you have to say, does the protocol work? You have to demonstrate that. And then you have to make sure that the people are actually following that protocol. And believe me, that COVID testing lab with 800 staff, anything that could go wrong did go wrong, including one group of 12 people decided, oh, I'm going to do this tweak here. And we could actually see their data shift because they thought they were making an improvement, but they're doing it in an uncontrolled fashion. All right. Equipment. You always have to demonstrate that the equipment, one, it's working, it's being well maintained, so that you can have confidence. Samples that come in on Tuesday, different samples come in 6 months later, the equipment is still working, it hasn't run down, or what have you. Reagents. So it's very easy to assume, oh, I purchased this reagent from Merck, or I purchased this reagent from Azenta, and they're manufacturers that we've already approved to use. But the thing is that they're also human beings. So all of a sudden they can send you a lot number, a particular shipment that maybe it did not meet their specifications, and all of a sudden we throw 600 precious samples on it, and due to the reagent lot number, it crashes. So what we usually do is we validate any new lot numbers before they so-called go into the clinical laboratory. And software. IT people, they love to make their software better and better. But it's very scary if they release a software and there's a bug in it and then your whole system crashes. And then with the people, is there accountability? Do people know who is responsible for what? Who can they go to for help? And then also, most critically, is the staff properly trained? There have been times, especially with that COVID testing lab, because of staff shortages, we had to shove people in there, and that really affected, before they were properly trained, that affected, a certain set of results. So trying to do whatever you can to minimize these type of sources of errors. And what it boils down to in a clinical lab setting, document, document, document. The simplest way to say it is if it's not written, if it's not documented, it did not happen. And you have no evidence that you are trying to do whatever it takes to minimize error. So I had gone into more details about the exam, and now I'm talking about the different stages of a laboratory process in a clinical setting. So I talked about the exam, or the analytical phase, which is you actually take your raw samples and you generate laboratory data. In reality, a test is more than just what happens in the lab. A test is pre-exam stage, which is basically, can't really see it, pre-exam stage, which is the patient gets hooked up to or is linked to a sample. So a patient doesn't come into your laboratory. A patient submits a sample. It could be blood, it could be a tumor, or what have you. And the first thing you want to do is really, how well did you track that the sample came from the patient, that they don't get uncoupled, okay? And to maintain as much privacy as possible, when a sample comes through a laboratory, usually a laboratory assigns a sample ID to that sample. So we do not see, Hamid's tumor is coming through. We would never see his name in the laboratory. It'll be X5, 931, and which is how it should be. So we try to maintain privacy, but because of that, you really have to have very secure conditions to link the right sample back to the right human being, all right? And so that's why, the things that you track, is it the correct patient? Are they ordering the correct test? Almost all laboratories have more than one test going through. It's very common for a molecular lab to have like 100 different tests coming through. So you want to make sure that when that sample is going through, it's going through the right test, the test that the doctor ordered, Did the sample arrive at the laboratory in the right condition? So it could be a courier, or it could be FedEx, because the sample is coming from the United States all the way to the United Kingdom. So we have so-called acceptance criteria. When the sample arrives at the laboratory, did it get crushed? Did a truck drive over it? Did it sit on a loading dock in Arizona and get baked to 90 degrees? Things like that. So these are the type of things that you actually have to ask at the pre-exam stage. So this is all the stuff that needs to be tracked before it even hits the lab. Goes through the lab, and then the post-exam stage. You have the laboratory results. So it could be, at this genomic position, we found this type of variant, or KRAS mutation. That's a result. But if you just told a doctor you have KRAS mutation such and such, many doctors don't really know what it is. So what you actually send back to the doctor or the clinic is the report, taking the laboratory result and actually giving them an interpretation of it so it is understandable for the doctor or the clinic or the hospital or what have you. So correct result, this is all in the post-exam. Correct result, is it going back to the correct patient? Is the report understandable? And then does the report go back to the right client? Okay, so like I said before, document every single step of the process. And this all falls under what's generally called a quality system. You try to maintain a very high quality. And then the thinking is you minimize errors. We're all human beings, and that's the great thing about these accreditations, these quality systems. It acknowledges errors will happen. So you track the errors, you try to learn from the errors, and you try to do everything you can to always improve your process. So, and then it goes back to minimizing errors. So again, this type of thing, I'm kind of trying to show the difference between the research setting and the clinical setting. And what I'm showing here is very often a laboratory, they have the so-called assay that is live, the test where they are handling clinical samples, but they're always thinking ahead to the future. What's going to be the next test that's coming out of academia that we will want to provide for our patients? And that's in the research and development phase. If the group is large enough, you have two separate groups. One group who's doing the research and development to help develop the tests, validate the tests for the future, and then you have your production group. They're the ones that's handling the real live samples. And so that's what all these arrows are just kind of showing you. There are many different activities that need to be done. It's a challenge. For example, I'm working for a very small biotech company. Currently, there's 5 of us lab people, and I'm not physically in the lab. And we're being asked by the business leaders, do the R&D, help develop the new tests. And meanwhile, I'm yelling, we need to get one test out the door, make it go live. It's a challenge when you have limited staff. But basically, the research and development is install, work out what's going on in the wet lab, work out what's going on in terms of software. You verify, you validate, and you also make sure you have enough staff to support all the activities that's required. So here is steps to accreditation. Who does the work? In a nutshell, everybody is involved in the work, all right? So it's not like you hire a quality manager and they do all the paperwork and then submit it to UCAS for approval or what have you. It's a philosophy. So every single staff member, from the person who's unpacking the boxes, they are all part of this quality system. And that's why I ask everybody, did you document that? Did you document this? Because otherwise I have no proof that you're even doing what you say you're doing. All right, so that's what it just keeps emphasizing, document, document, document. You have teams of people, the lab director, usually you need to have a nominal head, somebody who bears the ultimate responsibility of what's going on in the lab. You have a team of quality assurance, because this documentation takes a lot of time. And then, of course, everybody in the company is involved in that whole quality assurance. And usually what happens is if it's a new laboratory, they submit to UCAS, we want to be recognized as having a good, proper quality system in place. And there are 2 parts of that submission. One is the laboratory in its entirety, the staff, the quality management, basically Bible that got written up that everybody says they're going to follow. And then you also submit to UCAS that one test that you want to get approved. And then UCAS would basically says, yes, this laboratory is recognized, it's ISO 15189, and test number 1 is also recognized. And then as the lab continues on and wants to add to its menu, then you submit packages more specific for that new test. So eventually you have a menu of maybe 10 or 100 different validated tests. So the thing that must be remembered, the cost of quality is real. It does take money. It will take up your budget. And this is actually a slide that I kind of modified that came from the cost of quality in the automotive industry. That was the original source of the slide, because it's the same type of thing. Whether you're making a new vaccine or you're doing a new genetic test for cancer, or you're making a car that you don't want that car to fall apart on the individual who bought it. All these industries have quality systems in place. And what I'm trying capture here is that one, you could take the approach, screw quality. I don't want to spend any extra money. I just want to make my cars. I just want to run that cancer test. So you can go down to zero quality cost, but then you're at much higher risk of failure, all right? And so failure costs, it could be loss of samples, it could be loss of reagents, it could be the loss of equipment that's failed, it could be the loss of your reputation when you get sued for that, bad result, or your car starts exploding all over the place. So the failure costs would be very high. Then you can go for, okay, I don't want any failure. I don't want any failure. I'm going to do everything I can. And then the prevention costs, paying for the service plans, paying for having enough staff or extra staff so you never have a staff shortage. That could be killing for whether it's a clinical lab run by a hospital, or it could be too much for a biotech company or a manufacturing company. So obviously, you try to go for what's tolerable. What can you afford while minimizing as much risk as you can? So you try to go for that optimal quality level. And this is the thing that I have to keep reminding people when I go join a lab. You're paying for this. Don't just say, I got somebody to print out a whole bunch of papers and we're done. The whole laboratory runs on this, okay? And then, oh yeah, so I just kind of mentioned what are the different types of costs in terms of failure and stuff. So, okay, so why bother with it? We're all human. And that's the fantastic thing about having a quality system. It recognizes that we're all human. So what do we do to ensure the best for our patient? Because that is what really matters, all right? So you continually improve your whole quality system. You never assume, I'm done, it's perfect. It's not like that. We're all human beings. You demonstrate your competence. You enhance your efficiency, as you can imagine. And this really came in big when your laboratory is successful and you're processing 10 samples and you become so successful that you have 100,000 samples coming through at a time. You enhance your efficiencies. Financial advantages, of course. I talked about that cost of getting sued or litigation or fines from the government. Meeting regulatory payer requirements, and of course, public trust and reputation. And I had done a very quick search, and there is the Medical Laboratory Science Council of Nigeria, and I assume every African nation, or basically every nation, has their own way of monitoring the quality of the health work that's being done in their lab. So that is basically what I kind of wanted to emphasize. So I talked about it from a laboratory diagnostic setting, but I do want to remind you, this is something that would be part of anything that gets translated. Whether you're making a new therapeutic that you wanted to go out. At one point, you wanted to get translated, whether it's by a manufacturer or by a laboratory that will be synthesizing, your new compound or what have you. They need to be following this quality system. So I think that's basically it. So feel free to ask any questions, and I'll be around for the rest of the week and be happy to chat. Right.
We have comments or questions from the house. Who would go first? Comments or questions from the house? Who wants to go first?
Stunned you. No.
Thank you very much. That was a fantastic talk.
Well, thank you.
It's always great to see what happens in the clinical setting, which is a much more controlled environment.
Yeah.
But I wonder if we can start incorporating this thing even at basic research level. So what would you encourage laboratories to do in terms of ensuring that when one generation of PhD comes, the protocol and the system does not end with them? and then you have to start retraining because it happens most times.
Right.
Then you have to start digging out, can you send an email to somebody who graduated 2 years ago who will probably be on the beach enjoying and say, how did you figure this out? I don't want to receive such emails personally.
Right.
Yeah, so how do you encourage people in
Uh-huh.
research settings to also incorporate a controlled system as well?
Right. So like I mentioned, many of the companies have a research and development group. They all came out of the academic setting. And you kind of mentioned the specific, the perfect example. A new person joins the lab and it's like, you know, oh, the person who developed the assay that I'm supposed to work on has already left the lab. It's back down to the simplest way to do it is document it. Have that protocol sitting there. Classic thing is document, where are the samples? You know, have that spreadsheet that says this sample came from this source, this date, you know, its background, what freezer it's in. So it's basically the same thing. If you document it at the academic laboratory stage, you start developing the good habits. And then it makes it very easy to transition or hand it off to that new graduate student or even that new technician. If you can at least start off by giving them, here it is, here's a whole bunch of files. Ideally, you know, of course they would overlap, and then they could ask questions. What's the meaning of the data in this column? That kind of stuff. But it's the same thing. Documenting it is much easier than saying it's in my head, or what I see constantly, it's in my email. Yeah.
Any other comments? Okay.
So one of the reasons I was very excited to have you speak here is you have a unique perspective on academic careers versus industry slash clinical testing and so on. Can you speak to what the differences are and when these folks are making decisions about where they want to go in their careers, what are the sorts of things they think about?
Yeah, so I'm going to not answer your question, and it's because I'm already slotted to give a talk about careers.
Yeah.
So yeah. So, and then of course I'm always happy to talk about that while having tea or you know what have you. But yeah.
Thank you for your presentation, Yumi. So I will comment that your speaking English was very clear, better than Professor Ross English. I think they can testify. It was very, very clear. Please, I want to ask, you know, in academic setting, we carry out our research, maybe we observe a new compound, as Professor Ross said yesterday, that until it gets a past clinical trial, it's still a compound and not drug. Now, how can we move from being a compound to drug? How can the academia maybe collaborate with the clinical personnel or people in the clinic now to confirm our research in the lab and take it outside?
Yeah, so that's the classic question of how do you translate your findings to something so-called, I wouldn't say more practical, but more clinical, right? You caught it right there, you said it, collaborate, right? So try to find like-minded scientists who's interested, who could potentially be interested in your compound because they are doing clinical trials focused on that so-called disease target, right? So unfortunately, fortunately, it is a matter of networking and making that communication and meeting the scientists at the meeting, you know, getting them to see your face. Here's my data, you know, and it's like, oh my God, you know, we're looking for something like that to help support our clinical trial, or this is the disease area focus that I'm interested in. And it really helps to so-called be at the ground level with them, you know. So unfortunately, there is no single correct answer. It's a network is basically it. And there are multiple ways to get to your goal. And it's because every compound is unique, every disease target is unique. So yeah, it is a challenge to figure out what's the fastest way to get there. Yeah.
Line. The person said, do you always have quality control samples for each batch of samples you analyze? And do you do a batch effect when there is a variation per batch?
Perfect. Yes, 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 yes. So
Thank
for
you.
example, the company that I'm working with now, they want to get a new assay up and running. And it is run on a 96 well plate. The first column is all positive controls, and I kept insisting, give me a negative control, because that is one way of checking, are my reagents clean? If the negative control, water, gives me a result, I have to throw out the whole batch, because I cannot trust what happened to those 80 precious samples that are on that plate. So yeah, you always have controls for every single batch. And then ideally, you have controls and measurements for every single sample. So in other words, did I generate enough data so that my result I can have high confidence in? So there are times that the whole batch worked, the positive controls worked, the negative control was negative, and everything looks fine. But you have two samples in there as like, I don't think I generated enough data. I don't want to give them something that I don't have confidence. Those samples would fail. So yes, in other words, that is a continual process. Mhm.
Thank you very much for the presentation. So clear. There is this disconnect between academicians and the hospital in Nigeria. In your presentation, I see where you said you get samples from the hospital to the university, and it was analyzed and back to the patient and the doctor again. So this disconnect we have, what advice can you give us, the academicians, the universities, to maybe something to do differently that can connect us with the hospital? Or
Yeah.
what can we, yeah, that's the
Yeah.
question.
Yeah, exactly. That is not unique to Nigeria. It is unique all over the world. So when I was at University of Glasgow, before I got my position with this current biotech company, they had that exact problem. You had your scientists in the Cancer Research Center building beautiful assays. And they literally took one of their assays, it happened to be a next generation sequencing cancer panel, and they so-called threw it over the wall to the NHS clinical lab. And the NHS clinical lab says, where's the documentation? How do we validate? They were not communicating. All right. So the clinical world had their expectations, and the academic world had basically their results. And so the University of Glasgow tried to help reduce that wall by building a laboratory that was an alliance between, literally, it had staff from both the University of Glasgow side and the staff from the NHS clinical testing lab. And that way, when the assay got built, it got built in the controlled setting so that the moment the assay got approved by UCAS, the NHS lab says, oh, we know how it's built. We can run with it immediately. That took money, and that effort shut down, okay? So it's not trivial, but that's the type of thinking, is like maybe to have a formal collaboration with your counterparts in the clinical lab. What is it that you need in order to get this test to work? Well, actually, the real question is, do they want your test? Does it actually meet their needs, you know? And so collaboration, again, networking, collaboration, find your clinical counterpart, find that laboratory that's actually doing the accredited work and work with them. So, but yeah, it's a challenge.
Thank you so much.
Sure. Thank you.